Howdy, today on Flippin Science we're looking at indicators and acid classification. So science understandings we're going to look at. Acid-base indicators are weak acids or bases, where the acidic form is a different colour from the basic form. And acids can be classified as mono or polyprotic, depending on the number of protons. And then that can be broken up into mono, di, and triprotic. So as I just said, indicators are weak acids or bases that change colour at different pHs. And we haven't really talked about it yet, but a pH is a measure of how acidic or basic a solution is. They're handy because they can tell us that a solution is acidic or basic or even neutral depending on the indicator, and sometimes even mix of indicators, so you can get a wide variety of colours that show you the different pHs of solutions. So look at an example here with phenolphthalein, and I'd like to thank Rhys Lewis for this animation. So as we play the animation, we're changing the pH. We can see we've got hydronium ions there. Water in low pHs turns into hydronium ions because of the hydrogen ions. As the pH increases, we turn over to hydroxide ions being present in the water. As that happens, we get a change in some of the bonds on the phenolphthalein molecule. And when that happens, we get a bit of a change in shape. We get a slight change in shape there. That change in shape leads to a change in the way that light reflects off of the phenolphthalein molecule. In this case, it reflects wavelengths of light that make up pink in our brain. When we reverse the pH, where we go back to being an acid, the molecule changes shape back, and we get back to the colourless situation we were in. So with phenolphthalein, when it's in acidic up to about a pH of 8 or 9, it's colourless. So here we can see over here we've got a picture of the acid form where it's colourless, and over here where you can see the basic form where it's pink. That subtle change in shape leads to the change in the way that light reflects off it, and that changes its colour. So when phenolphthalein is colourless, it's in an acidic solution. When it's pink, it's in a strong basic solution. But that change depends on a specific pH, and we're going to see that with the indicators in a second. So on this page, we've got a wide variety of indicators, and we can see that for some of these indicators, the pH that they change at, there can be a bit of a range over which they change. Look at bromothiamyl blue, for example. In acidic conditions, it's a yellow. In basic conditions, it's blue. So when it's about neutral, it's around about a green colour, which is halfway between the yellow and the blue. So here I've got a few indicators and the pH ranges over which they change. So we had phenolphthalein before. So you can see it's colourless when it's at low pH, when it's acidic. But then when we get to the pH range of 8 or 9, that's when it starts to change over to the deep pink colour. Down here we have litmus. Litmus can be paper. Litmus is a substance. I'm pretty sure it comes from a plant, but don't quote me on that. There's two types of paper you can get, red litmus and blue litmus. Red litmus, if you expose it to a base, it turns blue. Blue litmus, if you expose it to an acid, turns red. But if you expose red litmus to an acid, it stays red. And if you expose blue litmus to a base, it stays blue. So it's a quick and easy way to test if something is an acid or a base. Here we have universal indicator, and it's shown up here as well. Universal indicator is a mix of indicators. So what happens is you can see a change in the pH all the way through the pH scale up to around about 12 or so. So when it's acidic, you get this kind of red colour. When we start heading towards neutral from the acid range, we get this kind of yellow. At neutral, it's a green colour, and then as you turn basic, you start to get more and more blue added, until when it's strongly basic, it's a deep purple colour. So here's a wide variety of indicators, and they're all themselves weak acids or bases. And when you're changing the pH, you're changing their structure slightly, and that leads to a change in their colour. So now we're going to talk about the classifications of acid. We can break acids up into two groups. There's the monoprotic and the polyprotic. Mono means one, protic means proton, so one proton. Poly means many, protic again means proton, so many protons. So monoprotic acid donates only one proton, whereas polyprotic acids can donate 2 or more protons. We further break up the polyprotic acids into di and triprotic, so we'll look at that on the next slide. So let's have a look at the monoprotic acids to start with, then we'll look at the polyprotic acids down here. If we have a look at this nitric acid molecule here, we can tell it's nitric acid. It's got a nitrogen there, 3 oxygens and a hydrogen, so HNO3. The reason why it's an acid is because this hydrogen atom here breaks off and that produces the proton. Now remember, acids are proton donors. Now nitric acid only has this one hydrogen here that it can donate. That's why it's a monoprotic acid. It's only donating one hydrogen. If we look at acetic acid over here, ethanoic acid, so CH3COOH, the hydrogen that breaks off is this one here. Not these ones attached to the carbon, they're attached fairly strongly. It's this oxygen-hydrogen bond where this hydrogen can break off. So again, this is monoprotic. This hydrogen here is the one that breaks off. Now, the polyprotic acids, like I said, there's diprotic, triprotic, and theoretically you could have tetraprotic, which is we've got 4 protons that can break off. There you're looking for 2 unstable hydrogens that can break off. So here we've got sulfuric acid. So we've got these 2 OHs attached to the sulfur, and then we've got double bonds between the sulfur and these 2 oxygens. What happens is this hydrogen and this hydrogen, again we've got that unstable bond between the oxygen and the hydrogen, they're the hydrogens that can break off. When that happens, you get a sulfate ion left over, SO42-. So because it can donate 2 protons, that makes it diprotic. Di means 2. If we look at tartaric acid over here, we can see we've got 2 carboxylic acid groups at the end of the chain, and we've got 2 hydroxyl groups over here. Now, for these ones, the hydrogens aren't breaking off of the hydroxyl groups here, they're breaking off of the carboxylic acid groups, and that's why they're an acid, it's carboxylic acid. So the hydrogens break off of the carboxylic acid part. Again, we've got 2 there, so it can donate 2. If we look at triprotic acids, so tri means 3, so they can donate 3 protons. So let's have a look at an example. We've got citric acid over here. So citric acid is a tricarboxylic acid. There's 3 carboxylic acid groups there. So this hydrogen, this hydrogen, and this hydrogen, those 3 break off, and that makes it triprotic. So now I've got a couple of questions for you. So look at this acid here. I want you to classify it as either mono, di, or triprotic. My second question is, a solution is a pH 10, methyl orange indicator is added, what colour would that solution be? Right, so let's have a look at the structure here. We've got an amine group here. These can be basic, which is kind of interesting. But we're asking what kind of acid this is. We've got a hydrogen here and a hydrogen here. We've got 2 carboxylic acid groups here. So there's 2 hydrogens there, so this is going to be a diprotic acid. If we look at the second question, solution is pH 10. If we look at methyl orange, we find pH 10. Line it up, we're going to get an orangey, yellowy colour in fact. It's a yellow-orange, closer to orange probably than yellow in real life. If the pH is very low for methyl orange, it's a deep pink colour. Methyl orange is a handy indicator. We use it often in titrations. I'm going to do some titrations later on this year. So today on Flipping Science, we looked at indicators and their uses, and we also looked at classifying acids. That's it, Flipping Science today. See ya.